Dear students, in this session we will be discussing about the characteristic features of viruses. In the previous sessions we have already discussed about so many features of viruses. We have discussed what is a virus, then about virion, about viroid, then also you studied about prions. So many things we studied. This viroid and prions, they are not viruses, but virion is the viral particle which is having all components to infect a host cell. All those things we already discussed. So again we are just brushing up. What is a virus? It is a submicroscopic organism. We cannot see it with the microscope. It is parasitic. They are obligatory intracellular parasites. They can divide only inside a host cell. They are filterable. The description was given in earlier sessions. What is meant by filterable organisms? Then they are just a nucleic acid surrounded by a protein coat. What is a nucleic acid? It may be RNA or DNA. RNA and DNA are known as nucleic acids. So they are just as nucleic acid surrounded by a protein coat. They will be having a coat which is made up of protein which is covering this nucleic acid. Nucleic acid is the genome of the organism. Then they are infectious agents. Why? They are obligatory intracellular parasites. So they will be infectious also because they require a host cell to multiply. They get into the host cell, hijack the host metabolic machinery and make the viral products, and later they assemble together into complete viral particles known as virion, and the cell is bursted, lysis of the cell occurs, and these virions come outside. They are capable of infecting other cells and they infect other cells. And they are acellular organisms. They do not have a cell. They do not have the features of a cell. They do not have a cytoplasm or cell wall or cell membrane likewise. Sometimes there will be an outer membrane, but we cannot compare that membrane with the cell membrane. For certain viruses there will be a membrane outside, and most often this membrane is acquired from the host cell itself, and they have a simple pattern of reproduction, and they are having different types of morphology for different viruses. So you can see here different morphology. These are all viruses and they are having different morphology. HIV virus, hepatitis, Ebola virus, adenovirus, influenza, rabies. These things also about the morphology also we discussed in earlier sessions. And these are some examples of viruses: human parvovirus, human polyoma virus, human papilloma virus, human adenovirus, variola virus, vaccinia virus, hepatitis B virus, varicella virus, cytomegalovirus, rhinovirus, hepatitis A virus, hepatitis C virus, rubella virus, coronavirus, hepatitis D virus, rabies virus, measles virus, mumps virus, Ebola virus, influenza virus, Bunyan-Vara virus, human immunodeficiency virus, Lassa virus, rotavirus, A, B, C, D, E. So these are all viral species. Okay, we already discussed about the taxonomy in the classification and taxonomy we discussed that viral species, the name is given by the disease, the name of the disease followed by virus. You can see rabies virus. Rabies is the disease and rabies virus is that particular species which is causing rabies. So one example is described here in a elaborated for you, that is parvovirus. Okay, this primate erythroparvovirus 1. Okay, it is generally called as parvovirus B19 or B19 virus or parvovirus B19, and the complete actual name is primate erythroparvovirus 1, and sometimes it is also referred to as erythrovirus B19. Okay, and it was the first known human virus in the family Parvoviridae. It was the first virus discovered in the family Parvoviridae, and its genus is Erythroparvovirus, and it measures around 23 to 26 nanometer in diameter. So this is a classification: family Parvoviridae, order unassigned. It is not assigned to any order, so it is unassigned. Scientific name is primate erythroparvovirus 1, and the higher classification is erythroparvovirus, and group which it belongs is group 2, single-stranded DNA virus. It is the genetic material is single-stranded DNA, and the rank, this parvovirus, that is a species of virus. Okay, parvovirus is a species of virus. And what is its role in disease? Parvovirus, it is causing fifth disease or erythema infectiosum. Okay, the disease which is caused by parvovirus is fifth disease or erythema infectiosum. It is one of the several expressions of this particular virus. Okay, this parvovirus B19 may cause several infectious expressions in the patients, but this is one of them. That is fifth disease or erythema infectiosum. And what is the main symptom? This is associated with bright red rash on the cheeks, and the rashes will be all over the body, but on the face it will be bright red in color on the cheeks, and it gives the disease the nickname slapped cheek syndrome. Okay, on the cheeks, these rashes will be bright red in color, so it appears like a slapped cheek. So it is known as slapped cheek syndrome. And at any age, this particular virus may infect humans, but the most common age group is 6 to 10 years children's who are coming between 6 to 10 years of age. Why it is known as fifth disease? It is so named because it was the fifth most common cause of a pink red infection associated with rash. Okay, there are so many diseases which is causing rashes over the body, red rash on the body, and we call it pink red infection. Sometimes it is referred to as pink red infection. Okay, this rashes, pink rashes all over the body, and this virus is the fifth common cause for the occurrence of this pink red infection. That's why it is known as fifth disease. And this virus is also associated with chronic anemia in individuals with AIDS, arthralgia, arthritis, aplastic crisis, hydrops fetalis. This is the electron micrograph of the virus, parvovirus in blood. This is the fifth disease. Child showing some signs of erythema infectiosum, also known as fifth disease. You can see the red rash, typical of this particular disease, and on the cheek it is more prominent. The red rash is bright red and it is more prominent. That's why it is known as slapped cheek syndrome. This is another picture showing the slapped cheek syndrome. Next, about the general characteristics of this virus. We will go in detail about the general characteristics of this viruses. So viruses can live only as intracellular parasites. They cannot divide otherwise. When outside a cell, they are inert. They won't divide, or they remain just like that as virion, complete viral particle. But they are capable of infecting other its host cell. So they are termed as obligatory intracellular parasites, and they usually target or infect cells which are capable of division. All the of all the viruses, they will be having host cells which are capable of division. Only then these viruses will be able to divide. So their host cell, they select the host cell who is capable of division, and these viruses, they may be present outside the cell or inside the cell. When inside the cell, they are virus, means they are active and they are dividing. But when they are present outside the cell, they are inert, and these inactive entities are known as virions. About the virion also we had a session earlier, and they range from 20 to 1000 mm in length. Not mm, nanometer. They range from 20 to 1000 nanometer in length. They are intracellular pathogens, and they are very small to be seen either by the naked eye or through an ordinary light microscope, compound light microscope. We cannot see with optical microscope these organisms. We can see only with the electron microscope. and these organisms are not considered as living cells unless they infect a cell. When they infect a cell, they are considered as living. When outside the cell, they are inactive entities, they are not considered as living. So they need the cellular and the genetic machinery of the host to multiply. Okay, they cannot multiply by their own, this virus. They require the cellular and the genetic machinery of the host to divide, to multiply, or for their metabolic activities. This is because their genetic material or their genomes, that is, it may be the RNA or the DNA, the nucleic acid present in the virus. This genetic material do not carry genes which are necessary for a metabolic activities. So they need to depend on the host genome to carry out the metabolic activities. So they cannot divide or multiply outside the specific host cell. And about the infection, these viruses can infect both prokaryotes as well as eukaryotes. So eukaryotes, there are so many eukaryotic cells, a vast number of host cells these viruses can infect. About the prokaryotes, they infect bacteria, and those viruses which can infect a bacteria are known as bacteriophages. And sometimes they are, and this what is the meaning of this bacteriophages? Bacteria eaters. Okay, so they infect bacteria. They bring about the lysis of bacteria. They grow inside bacterial cell. They attach to a bacterial cell, gain entry into the bacterial cell, divide, multiply inside the bacterial cell using the genome and the metabolic products of the bacteria, and they assemble into new virion particles. They then come outside by the lysis of the bacteria, or sometimes by budding they come outside, or most times there will be lysis of bacteria releasing these virions outside. And these viruses, they require the help of specific receptors present on the cell membrane to gain entry into the host cells. Only then it is with the help of the specific receptors on the cell membrane they gain entry into the host cell. That's why there is this specificity in the case of host cells. For a virus, there is its specific host cell. Virus cannot attack any, or for example, tobacco mosaic virus can attack, their host cell is this tobacco, the cells present in tobacco plant. They cannot infect other cells, animal cells. Okay, so each virus will be having its own specific host cell. That is because of the specific receptors present on the cell membrane which help them to gain entry into the host cell. They can bind only to the specific, with the help of the specific receptors, to the host cell. And once they are inside the cell, they usually insert and integrate into the genetic material of the host. That is what they are doing. The genome of this virus get integrated into the genome of the host cell. And while the host genome is replicating, the viral genome is also getting replicated. And also they use the molecular machinery of the host to generate various components required for the generation of viral particles. Okay, that is what this virus is doing. And the viral particles which are produced will be assembled later. That is, multiple copies of genome will be produced. Genome, that is the nucleic acid, RNA or DNA. So multiple copies will be produced. Also the protein codes, that will be also produced in multiple copies, and later they are assembled together to form the complete virus particle or the virion particle. The assembled viruses then come outside the cell and they are ready to infect other cells. And this emergence from the host cell may be by the lysis of the host cell or by budding from the host cell. They come outside. Okay, this is how generally how this virus work. Then differences between viruses and living cells. What are the difference? We told this virus, they cannot be considered as living entities, isn't it? So what are the differences between this virus and living cells? And they differ in mainly three ways. That is, one is simple and acellular organization. They are very simple, and they are not having a cell. They are not having a cellular organization. They are just a nucleic acid surrounded by a protein coat. Then the presence of either DNA or RNA as RNA, but not both in almost all virions. In the case of living cells, we see both DNA and RNA in a cell, isn't? DNA will be the genetic material, and RNA is produced from DNA for various reasons. But in the case of virus, we do not see RNA and DNA together. Either we see DNA or RNA. We do not see them together in a virus. Then the third thing is their inability to reproduce independent of the host cell. They can reproduce only inside the host cell. That is one difference. Then differences between viruses and prokaryotes. What are the differences between viruses and prokaryotes? Viruses do not have a defined plasma membrane. That is one difference. For prokaryotes like bacteria, there is a well-defined plasma membrane. But here for virus, there is no plasma membrane. I told you there may be a membranous coat, and that is always derived from the host cell. For certain viruses like HIV, there is a membranous coat around the virus, but that cannot be compared with the plasma membrane of the prokaryotes. Then they do not multiply by binary fission. That is another difference. Then they possess a DNA or RNA, but not both. In the case of prokaryotes also, we see both DNA and RNA in a single cell. They do not have the genes or biochemical mechanisms to produce ATP. That is another difference. They do not possess ribosomes. Okay. Then they are not sensitive to antibiotics. That is another difference from prokaryotes. Prokaryotes are sensitive to antibiotics, but these organisms are insensitive to antibiotics, because most antibiotics work by inhibiting the cell wall synthesis. Here there is no cell wall or not even plasma membrane, so they are insensitive to antibiotics. But they are sensitive to interferons, and these interferons are this bacteria are insensitive to interferons. These viruses are sensitive to interferons. What are these interferon? Interferon are a group of signaling proteins. Okay, they are a group of proteins produced by the host cell in response to viral infection. That is known as interferons. Okay, that is a way of what inhibiting these viruses. Okay, an immune reaction. Actually, it may be an immune considered as an immune reaction. That is, the cells produce a group of proteins in response to viral in response to the presence of viruses. These proteins are known as interferons. So that may be considered as a defense mechanism of the host cell. Then, since these viruses are depending on the host genes for most of its activities, that is for the replication, for the production of the coat, for everything, it is depending on the host cell. So they do not carry much genes in their genome. They do not carry genes for biochemical activities or for the formation of copies of the genomes. Such genes are not carried in their genome, and this will help to reduce the size of the genome, and so that it is a very compact, small and compact structure. That's why these viruses are this small, that is nanometer in size. They do not carry that genes for protein synthesis, ATP generation, nucleotide biosynthesis, etc. And for all these mechanisms, they depend on the host genome. And they carry only genes which is not available in the host genome. They carry only genes which are not available in the host genome, and this has enabled these viruses to make its genome very small. So once inside the host cell, they hijack the host genetic machinery and force the host cell to produce viral components. Okay, viral, whatever the virus require, that is made by the host cell. And when sufficient quantities of these viral components are produced, they will get assembled into complete viral particles or the virions, and it is released outside, as I told earlier. Then about the structural, we will move into the structural features of a virus. A complete viral particle is known as virion, that we already discussed about virion. And this virion, it is having two major components, that is the nucleic acid core and a protein coat. That's all this virion is having. Okay, virion is a viral particle which is capable of infecting a host cell. This is something which is, this is a particle which is outside the host cell. It is not inside the host cell, it is outside the host cell. It is capable of infecting a host cell. That is a virion. It is having a nucleic acid core and a protein coat. Nucleic acid core means that is the genome. Okay, nucleic acid, it can be RNA or DNA. And this RNA or DNA, it is surrounded by a protein coat. That's all the virus is having. And in certain cases, this protein coat will be covered by a membrane. Okay, in certain viruses like HIV, it will be covered by a membrane. Okay, that membrane is known as envelope. Some viruses having a surrounding lipid bilayer, that is known as an envelope. So for certain viruses like HIV, there will be an envelope covering the protein coat. Now about the virion, the structural details. So there is a nucleic acid core. Okay, that is the genetic material. There is the genetic material. So about the genetic material, it can be either DNA or RNA. We do not see them together. This DNA and RNA are not seen together. It will be either DNA or RNA. And this DNA and RNA strands may be positive or negative sense. Okay, they can be either positive sense or negative sense. Negative sense DNA or positive sense DNA, or positive sense DNA or negative sense RNA likewise. Okay, they can be positive, that is positive sense DNA or negative sense DNA, positive sense RNA or negative sense RNA. Then they can be single stranded or double stranded, like single stranded RNA or double stranded RNA, single stranded DNA or double stranded DNA. Okay, and they can be linear or circular in structure. But we do not find DNA and RNA together in a virus. Then there may be one or two molecules of the genetic material inside the capsid. One or two copies will be there of the genetic material. And introns are absent, altogether absent in viral genomes. Introns are non-coding sequences that we find in the genetic material. And these introns are altogether absent in viral genomes. And the nucleic acid molecule will be surrounded by specific proteins, forming a matrix. So this is about the genetic material. Next, about the capsid. The genetic material that we discussed earlier, and the matrix which is surrounding the genetic material, proteinaceous material which is surrounding the genetic material or the nucleic acid. These things are enclosed within a protein coat called the capsid. Okay, this protein coat is known as the capsid. And this capsid gives the characteristic shape to the virus. Okay, the shape of the virus is determined by the capsid this virus is having. Now the composition of capsid. This capsid is composed of subunits called capsomeres. It is composed of subunits called capsomeres. And this capsomeres, so there will be so many capsomeres joined together to form the capsid. And these capsomeres which have joined together to form the capsid may be identical, or it may be made up of a variety of capsomeres. Okay, in that case, there will be multi-subunit proteins forming the capsid. In the other case, when it is made up of same capsomeres, identical protein molecules make up the capsid. Okay, so capsomere may be made up of identical protein molecules or may be composed of a variety of multi-subunit proteins. And when multi-subunit proteins are there in the capsomeres, that usually render complex arrangements to the capsid. Okay, complex capsids will be made will be seen in those viruses which will be having multi-subunits proteins in the capsomeres. So this arrangement of proteins in the capsomeres, it is one of the identifying features of the virus. Okay, that is a certain the specific feature of the virus. This arrangement of proteins in capsomere that will be differing from virus to virus. This viruses, because of this arrangement of capsomeres in the capsid, the viruses can have different shapes. It can be polyhedral, it can be helical, or sometimes it can be complex forms, complex structures may be formed. Okay, polyhedral or many-sided, and that is commonly seen. That is a structure, is a shape we commonly seen in many viruses. Example, polyoma virus, picornavirus. Then helical. Helical form we seen in tobacco mosaic virus, rubella virus. Okay, in rubella virus we see it as hollow protein cylinders. Helical shape we see in tobacco mosaic virus. And these regular arrangements, that is the above mentioned arrangements, allows these viruses to be crystallized. Then there are also complex viruses. Here the capsid symmetry is not icosahedral or helical. They will be highly complex because of the arrangement of this capsomeres with the presence of multi-subunit proteins. And in such cases, the virus may be possessing tails or other structures such as additional walls surrounding the nucleic acids, etc. Okay, so example for complex viruses with complex shapes can be vaccinia virus, paramyxovirus of mumps, herpes virus, bacteriophages. Okay, so bacteriophages is a complex virus. It is having complex shape. It is not its shape is not that simple. It's we can say its shape is complex because of that capsid symmetry. Okay, bacteriophage that is described below. So next is about the nucleocapsid. Nucleocapsid is the genetic material together with the capsid. That is known as nucleocapsid. Okay, both together, the nucleic acid plus the capsid, we call it the nucleocapsid. Here the picornavirus, structure of picornavirus is shown, polyoma virus, then the tobacco mosaic virus, tubulovirus is shown here. Then here the capsid symmetry of vaccinia virus, paramyxovirus, herpes virus. These have complex symmetries, capsid symmetries. Next, about viral envelopes. So in several viruses, capsids are wrapped with a bilayered membranous cover, and these are known as envelopes. That is present in only certain viruses like HIV. This additional to the nucleic acid core and protein core, they are having one more structure in them, that is bilayered membranous covering, and that is known as the envelope. And these viruses are known as enveloped viruses. And many viruses that infect animals are enveloped viruses. Examples are HIV, human influenza virus, herpes virus, rhabdovirus. These are all enveloped viruses. And this bilayered envelope is acquired from the host cell itself. Okay, it is acquired from the host cell itself. That is, while this virus comes out of the host cell by budding, they will carry membrane along with them, membrane of the host cell along with them. And these envelopes carry glycoprotein complexes. These envelopes will be having projecting outside glycoprotein complexes called spikes. And these spikes help these viruses in attachment to the host cells. Also, one another advantage of the presence of this envelope is that they help these viruses they shield from the degradation by the host enzymes, because this is same as that of the host membrane. This envelope, the structure is this envelope is from the host membrane itself, so it is very similar to the host membrane. So it won't be degraded by the host enzymes. Okay. Also, it can evade immune attacks mounted by the host, because when the host immune system searches, when it finds this envelope, it cannot recognize this particle as a foreign particle because it is covered by an envelope which is very similar to the plasma membrane of the host cell. So it cannot identify that particle as a foreign object. That's how this virus evade the immune system of the host. Also, the presence of the envelope facilitates smooth entry into the host cell. It can very easily fuse into the plasma membrane of the host cell. But one disadvantage is that they degrade quickly. Okay, they degrade quickly. This membrane, we know our plasma membranes degrade quickly in situations like desiccation, changes in pH, heating, treatment with detergents, etc. So in such situations, this virus will also degrade. Okay, this viral envelope will also degrade, and since it is part of that virus, once this viral envelope is degraded, this virus cannot gain entry into the host cell. Okay, so such conditions, that is a disadvantage. That is a disadvantage to these viruses. That is one the other advantages are described above. And viruses like bacteriophages have more complex structures in addition to capsid. They are having a sheath, tail fibers, basal plate. Okay, and about bacteriophages we will be discussing in another session. Thank you.